All right, welcome back, fellow scientists. So today we're going to talk about the biotechnology process of cloning. So before we talk about cloning, we need to define what a clone is. So our definition that we're going to use for a clone, we're going to say that a clone or clones are organisms with the exact same DNA. Organisms with the exact same DNA. So, do clones occur naturally? Yeah, any organism that reproduces asexually, they're producing clones of themselves. Do we ever have any human clones? Yeah, identical twins. They have the exact same DNA. They came from the exact same egg. If you remember back to when we were talking about stem cells and we had the morula, we said that if one cell breaks off from the morula, then we have identical twins. So cloning is not something that is totally unnatural and totally out of the realm of possibility. But humans directing cloning, that's something new. That's something that we really have to take a hard look at. So we're going to start we're going to use the example of Dolly the sheep. To look at cloning. Dolly the sheep, it was announced in 1997 that this was the first mammal to be cloned. Is they named her Dolly the sheep. So I'm going to use this diagram. I'm kind of cheating. Normally I would draw the sheep, but I don't want to suffer through that. So in your notes, if you could try to reproduce this diagram, just generally. And for those of you that are not artistic like I am, if I were drawing this, here's how I would draw sheep. So I would start with a cloud, and then I would give the cloud legs, and then I would put a small little cloud for a tail, and then I would draw an oval, and then I would draw another little oval for an ear, and then we'll make it a happy sheep. So anyways, that is my attempt at drawing a sheep, which is why I am using this diagram. So I apologize that you had to suffer through that, but if you're not artistic like me, feel free to use that technique to draw your sheep in your notes. So first things first, we'll number these sheep. So we'll say this is number 1, number 2, number 3, and number 4. So the way that we start the process of cloning is we take a regular diploid cell, a regular somatic cell, from sheep number 1. Now sheep number 1, it could be a female or it could be a male. It doesn't matter. All it matters is that we have a diploid cell with a diploid nucleus. Remember, diploid means 2 sets of chromosomes. Every organism that is produced through sexual reproduction gets 1 set of chromosomes from mom, 1 set of chromosomes from dad. Sheep are no exception. So, diploid nucleus. Then we would take an egg cell from sheep number 2, and because we're taking an egg cell, obviously this sheep has to be female. Now, if you'll recall, egg cell is a type of gamete, and that is haploid. It only has 1 set of chromosomes that's going to normally pass on to the next generation. Well, not so with cloning. What we do with this egg cell is we take out the haploid nucleus. So that goes into the garbage, right? And so now we have this empty egg. So what scientists do is they take this diploid cell and they take this empty egg and they fuse the two together with a very small electric shock. Right, so this gray circle that represents the cytoplasm and all the organelles of the original cell, that's no more. That is either absorbed into the egg or it's just kind of waste that goes in the trash. But now the main thing is we have a diploid nucleus inside an egg. So this is a diploid 2N egg. Now the normal way that we get that is through the process of fertilization. 1 set of chromosomes from dad meets 1 set of chromosomes from mom, and then we have an egg with 2 sets of chromosomes. The only difference is that this egg, the DNA for this egg, came all from sheep number 1. It didn't come from a combination of 2 different sheep. So we take this diploid egg, and because it's diploid, the cell recognizes that, and so it says, oh, I must have been fertilized. And so it starts dividing into an embryo. Then this embryo is implanted into the uterus of sheep number 3. Obviously, sheep number 3 has to be female. And so then sheep number 3 goes through the normal process of pregnancy, and after the appropriate gestational period, then sheep number 3 gives birth to sheep number 4. Sheep number 4 is, in our case, Dolly, right? Sheep number 4 is the clone. So, quiz question, which sheep is Dolly identical to? Which sheep is Dolly a clone to? Is it 1, 2, 3? I guess not 4, but 1 or 2 or 3? If you said 1, you're exactly correct, because all of Dolly's DNA, right, this embryo developed into Dolly, all of Dolly's DNA came from sheep number 1. So if sheep number 1 was a female, then sheep number 4 is going to be a female. If sheep number 1 was a male, then sheep number 4 is going to be a male. All right, so let's look at the fate of Dolly, because this is an interesting story as well. How far down? There it is. Okay, let's go down a little bit further. Sweet. All right, so she did live a healthy, happy, normal life. She gave birth to 6 lambs. Well, I guess I should qualify happy, healthy, normal life. She developed arthritis at about 5 years of age, which is kind of early for a sheep. And also she died from a disease known as sheep pulmonary adenomatosis, or SPA, in 2003. Excuse me. If you do the math, they announced it in 1997. I believe that they actually, sheep number 3 gave birth to Dolly in 1996. So if you do the math, she was 7 years old. The average lifespan for a sheep is about 11 years. So she got arthritis early and she died early, which leads some people to believe that there's a genetic factor that helps to determine age. So if this sheep was 5 years old, then it's almost as if this DNA has the genetic markers or is somehow some quality, some characteristic 5 years old, which makes this embryo 5 years old, which makes this lamb then 5 to 6 years old already when it's born. And so they're still doing research into that and why that happens and how to possibly fix that. But anyways, that's the general process of cloning. Now, here's some maybe more human applications for cloning. You guys remember talking about stem cells. So stem cell, you take the inner cell mass from an embryo and you culture it in a petri dish and then you incubate it. Excuse me, horrible. Then you incubate it with certain factors, and if you incubate it with the right factors, then it becomes heart tissue. If you incubate it with other factors, it becomes pancreatic tissue. If you incubate it with other factors, then it becomes ligament or muscle or whatever you want it to be. And we talked about the ethics of stem cells, and we talked about where they come from and from a Christian perspective. So remembering all of that, now here's a new twist on all of that. This is called therapeutic cloning, and this is more controversial than stem cell technology. This is just theoretical. This, to my knowledge, at least, is not even in practice or being researched at all. This is just an idea. So if you take, just like with the sheep, if you take an egg cell, if you remove the nucleus, and then if you implant or input a diploid nucleus from someone else, then you create a human embryo. What you can then do with that human embryo, exactly like we did with the stem cells, you can take out the inner cell mass, you can culture it, and you can get different types of tissues. Now the advantage so this is called therapeutic cloning, and the advantage that therapeutic cloning has over stem cell research is this inner cell mass is not genetically identical to this human being. However, if we take a cell from the person that needs the new heart or the new pancreas or the new ligament or something like that, and we implant that nucleus into the empty egg, then this embryo is going to be a clone, is going to have the exact same DNA as the person that donated the nucleus, as the person that needs the heart or the pancreas or the ligament. So then the person won't have to go on immunosuppressant drugs. And it'll be just like they have their own heart with their own DNA, because it is. So that's something that, again, we'll talk about in class, and we'll talk about the ethics and implications and ideas of therapeutic cloning. Until next time.